Groundbreaking CRISPR gene edit blocks malaria spread in mosquitoes. Imagine a world where mosquitoes, instead of being our enemies, become our allies in the fight against malaria. This isn't a distant dream, it's a reality that scientists are actively working towards. As mosquitoes develop resistance to insecticides and malaria parasites outmaneuver our frontline drugs, traditional public health tools are struggling to keep up in the battle against one of humanity's most lethal diseases. But here's where it gets controversial. What if we could edit a single amino acid in mosquitoes to stop them from transmitting the parasite? This is exactly what a groundbreaking study published in Nature has achieved. The study, titled Driving a Protective Allele of the Mosquito FREP1 Gene to Combat Malaria, introduces a novel genetic strategy to curb malaria transmission. This method doesn't involve eliminating mosquitoes or inserting foreign genes. Instead, it reshapes mosquito populations from within. The researchers have developed a system that includes a self-propagating, allelic drive, to spread the resistance trait through wild mosquito populations. Ethan Beer, Ph.D., a professor at the University of California, San Diego, and one of the study's authors, explains that this is a third genetic strategy to combat malaria. The first two methods are either suppressing the mosquito population or modifying it to produce transmission-blocking antibodies. This new approach simply restructures the existing population so that mosquitoes inherently resistant to malaria replace those that efficiently transmit the disease. The new method targets a mosquito gene called FREP1, which helps the insect feed on blood but also serves as a key gateway for Plasmodium parasites to reach the mosquito's salivary glands. By switching one amino acid in FREP1, from leucine, L224, to glutamine, Q224, the researchers created a naturally occurring variant that effectively halts parasite transmission without affecting mosquito viability. This means the mosquitoes can still reproduce and develop normally, but they can no longer transmit malaria. The idea that changing a single amino acid could render mosquitoes refractory to infection by malarial parasites was a real surprise to Beer. And the second surprise was that their drive system was more efficient than predicted. This efficiency is due to an effect wherein individuals that carry the gene cassette but not the preferred FREP1Q allele are dominantly eliminated as a byproduct of CRISPR activity. The allelic drive system developed by Beer's lab in collaboration with George Demopoulos, Ph.D., at Johns Hopkins University, employs a CRISPR-Cas9-based gene editing cassette. Unlike standard gene drives, which must remain embedded at the edited site, the allelic drive's components can be located elsewhere in the genome. This modular design allows for greater flexibility and, crucially, the ability to self-eliminate once its job is done. Beer explains that the gene cassette encoding the CRISPR components can be placed at a different genetic site than the one to be acted on. This means the gene drive must copy itself at the location at which it is inserted into the genome. For example, one can engineer the genetic system to disappear and just leave an altered population in which all individuals are inherently refractory to infection by malarial parasites. If one used a gene drive expressing transmission-blocking antibodies, that genetic element would have to remain in the population in order to exert its effect. The team linked the guide RNA, gRNA, targeting the non-preferred FREP1, L224 allele directly to the preferred Q224 variant. This design ensures that the gRNA is never inherited separately from the desired protective allele, avoiding the risk of propagating unwanted mutations. Beer told Jen that if such a separation happened, there would be a possibility that the drive system could copy an undesired mutant version of the gene, generated during the drive process, as well as the preferred allele, which could reduce the effect of the drive. The system they developed could not do that, and no such unwanted mutant alleles accumulated during the drive process in their experiments. Tests in Anopheles stephensi, a major malaria vector in Asia, showed that the single amino acid switch blocked two parasite species from reaching the mosquito's salivary glands. Importantly, the edited mosquitoes reproduced and developed normally. Demopoulos highlighted the beauty of this approach, saying, "With a single, precise tweak, we've turned it into a powerful shield that blocks multiple malaria parasite species." While the FREP1Q-linked allelic drive is not suitable for Aedes mosquitoes, which transmit dengue, the platform is broadly adaptable. Beer noted that the system is broadly generalizable to any situation in which one can generate efficient gene drives. The only requirement is that there are two functionally equivalent variants of a gene. Looking forward, Beer envisions integrating this strategy with traditional public health interventions for maximum impact. "We do not imagine that any genetic system would be a silver bullet," he said. "But combining suppression drives, modification drives, and allelic drives with tools like bed nets, insecticides, and antiparasitic drugs could create a much more powerful system." One particularly attractive combination would be first to suppress mosquito populations and then modify the remaining few such that they can no longer transmit the parasite. The project also illustrates the power of interdisciplinary, international collaboration. Beer's team worked closely with Demopolis Lab, FREP1 biology experts at Johns Hopkins, and mathematical modelers from the University of California, Berkeley, and the University of São Paulo. Demopolis emphasized that this breakthrough is the result of seamless teamwork and innovation across institutions. "Together, we've harnessed nature's own genetic tools to turn mosquitoes into allies against malaria." But here's the part most people miss: this approach raises ethical and ecological questions. Should we be altering wild populations in this way? What are the long-term effects on the ecosystem? These are questions that need to be addressed as we move forward. What do you think? Should we embrace this technology, or are there too many unknowns to proceed with caution? Genetic revolution: how mosquitoes are fighting malaria with a single mutation. Imagine a world where mosquitoes, the deadliest creatures on Earth, could no longer spread malaria, a disease that inflicted misery on millions. In 2023, these notorious blood-sucking insects were responsible for infecting approximately 263 million individuals with malaria, leading to nearly 600,000 fatalities, with a staggering 80% being children. This alarming statistic underscores the global health crisis at hand. Recent attempts to curb malaria transmission have hit significant roadblocks due to the development of resistance among mosquitoes to insecticides, as well as similar challenges faced by the malaria-causing parasites themselves in resisting medications. Adding to this complexity, the COVID-19 pandemic severely disrupted ongoing initiatives aimed at combating malaria. However, there is hope on the horizon. A collaborative research initiative involving scientists from the University of California, San Diego, Johns Hopkins University, UC Berkeley, and the University of São Paulo has led to a groundbreaking discovery that could revolutionize our approach to halting malaria transmission. Biologists Zhiqian Li and Ethan Beer from UC San Diego, along with Yue Dong and George Demopolis from Johns Hopkins University, have devised an innovative method using a CRISPR-based gene editing system. This technique meticulously alters a single molecule in the mosquito's genes, a seemingly minuscule adjustment that prohibits the transmission of malaria parasites. Notably, these genetically modified mosquitoes retain their ability to bite humans infected with malaria and pick up the parasites, but they are unable to transfer those parasites to others. The goal is to propagate this malaria-resistant trait through mosquito populations, eventually leading to a complete stop of disease transmission. As Professor Beer notes, changing just one amino acid in mosquitoes to a naturally occurring variant that prevents them from contracting malaria is nothing short of revolutionary. It's astounding how one small alteration can have such profound implications. This cutting-edge system employs CRISPR-Cas9, often likened to genetic scissors, along with a guide RNA to create an exact incision in the mosquito's genome. After making the cut, it replaces the harmful amino acid, which facilitates malaria transmission, with a protective variant. The targeted gene, known as FREP1, is crucial for mosquito development and their ability to feed on blood during bites. The innovative gene editing alters an amino acid in FREP1 from one identified as L224 to another referred to as Q224. This transformation effectively prevents disease-causing parasites from reaching the mosquito's salivary glands, thwarting their potential to infect humans or animals. In extensive studies, Professor Demopoulos and his lab evaluated various strains of Anopheles stephensi mosquitoes, recognized as major vectors for malaria transmission across Asia. Their findings confirmed that switching L224 to Q224 efficiently blocked multiple strains of malaria parasites from entering the salivary glands and, consequently, from spreading to others. The brilliance of this methodology lies in its utilization of a naturally occurring genetic variation in mosquitoes, elaborated Demopoulos. With a single, precise modification, we've created a formidable barrier against numerous malaria parasite species across various mosquito populations, facilitating flexible and practical strategies to combat this illness. In subsequent experiments, researchers discovered that while this genetic adjustment inhibited the parasite's ability to infect, the overall growth and reproductive capacity of the mosquitoes remained intact. Mosquitoes integrated with the new Q224 variant showed comparable vitality to those with the original L224, representing a significant achievement. This is crucial since the functionality of the FREP1 protein is vital for the mosquito's biology, apart from its role in facilitating malaria conveyance. The researchers have also pioneered a technique akin to a gene drive, enabling mosquito offspring to inherit the Q224 allele and propagate it within their populations, ultimately obstructing malaria transmission. This novel, allelic drive, concept is reminiscent of a recent groundbreaking system developed in the Beer lab, which successfully reversed insecticide resistance among crop pests. Previously, we crafted a self-erasing drive that transitioned a fruit fly population from insecticide resistance back to their naturally susceptible state, eventually vanishing and leaving behind a restored insect population, explained Beer. We could develop a similar disappearing drive system that transforms mosquito populations to carry the parasite-resistant FREP1Q variant. While the researchers have demonstrated the efficacy of the L224 to Q224 modification, they are still endeavoring to fully understand the mechanisms behind its success. Research continues on how the Q224 amino acid effectively obstructs the pathway through which the parasite infects the mosquito. This advancement is a result of exceptional teamwork and creativity across various institutions, remarked Demopoulos. Together, we have harnessed genetic tools from nature itself to transform mosquitoes into allies in the fight against malaria. This research not only sparks excitement but also raises questions about the implications of genetic engineering in nature. What do you think? Could we soon see a world free of malaria, or does the idea of genetically altering species evoke more concerns than it solves? Share your thoughts and join the conversation.